So, the in-gel digestion of proteins isolated by the gel electrophoresis remains a core area in the mass spectrometry or in any of the proteomics applications. So, the following video which we are going to watch is going to provide the broad guideline for the in-gel digestion. However, the recipe is very flexible and it varies from lab to lab to meet the specific requirements of particular proteomic experiment. The in-gel digestion procedure is compatible with the downstream mass spectrometry analysis, whether you want to continue with the MALDI-TOF or you want to do the LC-MS based mass spectrometry analysis. In-gel digestion of proteins for MS analysis video. Often one or two dimensional gel electrophoresis is applied for separation of complex mixtures of proteins prior to mass spectrometric analysis. In-gel proteolytic digestion of separated proteins is performed to cleave the proteins of interest present within the polyacrylamide matrix. You can see the overview of various steps involved in this process. One can use one dimensional electrophoresis or take the protein samples directly from the mass spectrometry based experiments, separate those on LDS page or 2D gel and then excise the band of interest or a spot of interest. The gel based techniques increases the dynamic range of analysis, since they involve sequential separation of proteins based on the molecular weight, lower to higher molecular weight. The mass spectrometric identification of the target protein greatly depends on the efficacy of the in-gel digestion process that generates mixture of peptides from the target protein through proteolytic digestion. This slide gives you the overview of various steps involved in the in-gel digestion process and also how various type of samples can be analyzed by using this method, whether you have 1D SDS page gel, 2D SDS page gel. In either way, you have to do the in-gel triptych digestion. Once the peptide fragments are generated, then those can be analyzed using LC-MS or MALDI-TOF or different type of hybrid mass spectrometry techniques. So, in-gel digestion, it is a multi-step procedure, but remains central to the proteomic applications. In-gel digestion includes a spot selection, a spot excision, stain removal, reduction, alkylation, proteolytic cleavage and finally, extraction of the peptides. So, let us say you have run a gel. First of all, you need to rinse the entire gel with water for few hours with intermittent changing of the water. Now, you would like to excise the band of interest or the spot of interest. So, keep a glass plate inside a laminar hood and clean the surface carefully. Excise protein spot with a clean sterile scalpel and place the gel slice into Eppendorf tube. It is possible you have excised the large spot or large band, then cut the slice into cubes while avoiding too small pieces as they can clog the pipette tips. So, this is the larger spot. You cannot directly take this one for doing the in-gel digestion. So, you need to excise that into small cubes. Now, keep these small gel pieces into a sterile microcentrifuge tube. Now, you can add 50 to 100 microliter of stain removal solution. For the large gel pieces, take enough liquid to cover it completely. So, you can adjust the volume depending on the size of your spots or the band. After addition of the stain removal solution, rotate it on a shaker for 30 minutes at room temperature for complete removal of the stain from the gel pieces. It is recommended that you change the solution after every 10 minutes. By changing the solutions and removing it after every 10 minutes, the Kumasi Brilliant Blue stained gel pieces becomes colorless. So, you have seen the animation. Now, once the staining is removed, then you can add dehydration solution. So, add 50 to 100 microliter of dehydration solution and rotate that on the room temperature. You need to ensure that you change the solutions after every 10 minutes, so that gel pieces becomes white and stick together. Now, spin the gel pieces down at 1000 G for 30 seconds. Once centrifugation is complete, then remove all the liquid. After removing the solution completely, then add reduction solution. So, add 30 to 50 microliter of reduction solution to completely cover the gel pieces. Incubate it 30 minutes at 56 degrees centigrade. Now, treatment of protein residues with dithiothriotol breaks the disulfide bonds. Now, chill down the tubes to room temperature, add 50 microliter of dehydration solution, mix it properly and incubate for 10 minutes. Now, remove all the liquids. Then add alkylation solution. So, add 30 to 50 microliters of alkylation solution and incubate it for 20 minutes at the room temperature in dark condition. The iodoestamide prevents the reformation of disulfide bonds. It is an alkylating agent. It adds iodoestamide group to the sulfhydryl group and prevents disulfide bond formation. Now, your incubation is over. So, you can remove the tubes and now remove the solution. Now, add 50 microliter of dehydration solution, mix it properly by vortexing and incubate it for 10 minutes. Again, you need to remove all the solution air dry the gel pieces and then add 25 microliter of trypsin solution, around 500 nanogram. So, add trypsin to the dry gel pieces and keep it on ice for 30 minutes for absorption of enzyme by the gel pieces. Add 25 microliter of ammonium bicarbonate buffer. It is same buffer in which trypsin is prepared, and incubate at 37 degrees for overnight 12 to 16 hours for proper proteolytic cleavage. So, prior to MS identification, proteins are digested to generate peptides. There are several proteolytic enzymes are available. Chymotrypsin, trypsin, pepsin are some of the enzymes commonly used for proteolysis. After the overnight step, you have to stop the reaction by keeping the reaction mixture in ice. Now, after the overnight incubation, the peptides generated through the proteolytic digestion can be extracted by using extraction buffer containing 0.1% formic acid or trifluoroacetic acid in 50% of acetonitrile solution. Now, collect the supernatant in the small aliquots and extracted samples can be stored in these small aliquots. The efficient extraction process is essential to ensure the release of peptides from the gel matrix to the solution. Now, all the solution volumes described in the protocols are recommended volumes, but depending upon the experimental requirement, you can optimize and change these volume and the incubation timing according to your experimental requirements. Now, these samples can be stored and used for the further mass spectrometry analysis. The in-gel digested protein samples are further processed by using zip tip pipette tips, which contains C18 or C4 media for enrichment of peptides prior to MS analysis. Zip tip pipette tip is a 10 microliter pipette tip with a bed of chromatography media fixed at its end. It is used for concentrating and purifying peptides as well as removing salts, detergents and interfering agents. So, first you need to attach the zip tip pipette tip on top of a suitable micropipette. Condition the zip tip with 10 microliter of acetonitrile. Perform the wash step 3 times and wash it with 0.1% trifluoroacetic acid or TFA. So, after watching this video, now you are very clear about the in-gel digestion process, how various steps are important to perform these experiments. Now, once you have done the in-gel digestion, you can directly use these triptych digest for further mass spectrometry analysis, but it is often recommended that in between you add one more step, which is sample cleanup. You do not want your columns or your MALDI instrument to get clogged due to the salt or some other interfering residues present in the mixture. So, it is recommended that one should use a cleanup step in between. So, the in-gel digested protein, they can be cleaned up by processing further using zip tip pipette tips, which contain C18 or C4 media for enrichment of the peptides. Salts and interfering agents, the detergents are washed, and finally, the samples can be eluted in a very small volume of the solvent. So, now you know how to perform the cleaning step by using zip tips. Now, you have the sample ready and you have selected the matrix. So, now let me show you this various steps involved before you can actually start the MALDI experiment. So, you need to select the matrix, you need to prepare a matrix, you have already done the sample purification. Now, sample need to be deposited on the MALDI plate. Either you can mix with the matrix or you can do these two separately. There are various combinations one can try, and then once both sample and matrix is deposited on the MALDI target plate, then you are ready to do the drying, and then plate can be used for MALDI-TOF instrument for further analysis. Let us first talk about matrix selection. So, the important step in MALDI-TOF analysis is selection of appropriate matrix for the sample. The matrix selection mostly depends on the molecular weight of the target to be analyzed, and often the type of application which you intend to do by using these instruments. So, these matrices are low molecular weight organic compound with low vapor pressure and volatile nature. Most of the matrices are acidic in nature, so it can easily excite the photon and ionize analyte for the analysis. However, there are few basic matrices are also available. In the slides, I am giving you an overview of few matrices and some of their properties, but there are many more properties which is not mentioned here, but just to give you certain major features of these matrices commonly used for the various applications. So, one is the alpha cyano 4 hydroxy cinnamonic acid. When you have peptides less than 5000 Daltons or lipids and nucleic acid, one can use this matrix. One can also use cinnapinic acid if peptides and proteins are having more than 5000 Daltons, and it can also be sometimes used for the lipids. Then you have options such as 2,5 dihydroxybenzoic acid, also known as DHB, small molecules and peptides which are not ionized by the other molecules can be analyzed by using this matrix. Trihydroxy acetophenone THAP, this is used for small nucleotides and also used for phosphorylation and specialized applications. Then we have picolinic acid, which is generally used for the nucleotides. So, these are only few representative matrices. As you can see, there are many options available for selecting the matrix depending on the molecular weight and the type of applications. But regardless of these matrices absorb energy from the laser source and converts both matrix and analyte into the gaseous phase. Matrix can also ionize analyte molecule by providing energy which comes from the laser bombardment. Now, once you have selected a matrix, matrix can be prepared by mixing it into a suitable solvent and vortex it for few minutes, so that it can dissolve properly. Now, you are ready with both your analyte, the protein which you want to analyze, as well as the matrix which you have selected for your application. Now, one need to think how to deposit that sample on the MALDI target plate. So, there are many ways of deposition of sample and matrix onto the MALDI plate. Mostly sample and matrix are mixed in a Appendorf tube, and then the mixture is directly deposited by using a micropipette onto the MALDI plate. But one can also try various combinations. In one approach, the sample is first deposited to the multiplate, followed by the matrix is deposited above it, and then it is properly mixed before drying process can happen. Other way of doing is to apply that with the sandwich based method, in which first a small amount of matrix is deposited on the plate, then you add the protein sample, and again the matrix is spotted on top of it. So, that you have enough matrix in the below and the above of the analyte. So, one can try different combination of placing the matrix and the analyte, and then once you have placed all of this sample of interest on the multiplate, then you are ready to dry the target plate. After spotting is done and multiplate is dried almost 30 minutes, then the instrument can be turned on and MS analysis can be performed. Now, there are various type of configuration of these instruments available, as well as there are various type of commercial software which help to operate the hardware. It is not possible to go into individual detail, but I am going to show you the generic steps in the following video of MALDI-TOF instrumentation. MALDI is performed in two steps. In first step, the compound for the analysis should be dissolved in a solvent containing small organic molecules known as matrix. This mixture is dried before analysis, and liquid solvent used in the preparation of solution is removed. So, in this video, by depicting the matrix preparation as well as instrumentation, we will try to give you overview of the MALDI-TOF instrumentation. So, spot the mixture on the multiplate. How uniformly you can plate these mixtures on the multiplate ensures your good spectra and data quality later on. Completed on the multiplates, the samples are allowed to dry for 30 minutes. After which the instrument is switched on and MS analysis can be performed. While these steps are happening, you need to ensure that instrument is on. So, click on the software and open the acquisition window, and then click on the open door. Insert MALDI target plate, face up with the cutoff corner to the front, and now by using software, close door. The door of insertion chamber is now closed. You can select the plate, you can view the overall plate on the screen, and then select the spot which you want to analyze. So, click on the yellow target in the acquisition window and select go to the location. You can now do the laser bombarding and peptide spectrum is generated. We have shown here one standard protein, bovine serum albumin. So, you have to look at various location where you can get best spectra from that spot, and then you can freeze it. Same process can be performed for different spots and different regions. Now, we have shown here a spectra for the PEP mix. So, now you are clear with how to perform the MALDI-TOF experiment. Now, let us add one more mass analyzer. So, now we have a configuration of MALDI-TOF-TOF. So, MALDI can be coupled to the tandem time of flight in combination with another time of flight. So, TOF-TOF or with hybrid quadruple time of flight analyzers, which are separated by the collision cells. Now, for proteomic application, it is recommended to use the TOF-TOF or Q-TOF. The peptide ions are accelerated through the first time of flight tube, as you can see in the slide, and then they are dissociated by introducing an inert gas into the collision cell. This process allows the collision induced dissociation spectra from the MALDI produced from the precursor ions. Now, these hybrid configurations are more sensitive than the triple quad and the single time of flight. So, the combination of TOF-TOF allows the protein identification through the peptide mass fingerprinting, and high throughput analysis of the protein or proteome is possible with the hybrid TOF analyzers.
Open the Matrix Science browser window to carry out online data analysis. For peptide analysis, click on Peptide Mass Fingerprinting. Mascot Peptide Mass Fingerprint. Please enter the user ID and details to acquire information in case of any network loss. The following parameters should be selected. Database. Primary databases include SwissProt and NCBI. Select SwissProt database for data analysis. Enzyme. Select enzyme as trypsin. Missed cleavages. Missed cleavages are allowed up to 1. Next is taxonomy. The protein extracted from the biological specimen has to be assigned to a particular species or a group of species to which the sample belongs. When you are not sure of organism, select all entries. Fixed modification. Select carbamidomethyl. Fixed modifications are applied collectively across the database to account for change in mass of specific residue. Now scroll down and select oxidation. These are mass changes suspected to occur during sample handling and accounted for by increasing the number of primary sequences compared against experimental masses. Include it as variable modification. Depending upon user needs, the parameters can be changed. Protein mass is the mass of intact protein and is optional. Set peptide tolerance as plus minus 1.2 Daltons. Mass values. It specifies the type of charge to be examined. Select MH positive. Select monoisotopic. Report top 10 hits. Select decoy for statistical analysis. Copy the M by Z values and paste in the selected box for mascot search. Click on search. Data output. The output can be seen in 3 sections. In section 1, the summary of set parameters defined by user. Section 2, mascot score histogram. The number of protein hits with score is plotted along the graph. Section 3, summary report in which matched proteins from the database with the details of important parameters are displayed either in concise format, protein format, and the data can be exported too. Data analysis. If the search parameters are not the best fit, the software generates an error message. Depending on the error message, user needs to change the parameter setting and do the search again. In section 2, Mascot score histogram, the number of protein hits and their score is displayed along the graph. Protein view section displays matching of the query peptide to the protein sequence in the database. The sequence type, the matched region, what is the expected and calculated values of the query peptide and sequence details. For better protein identification and to increase protein score, CID of each peak generated is carried out to generate MS-MS data. For such data analysis, MS-ION search option is selected from the Matrix Science browser window. In MS-MS search tool, more input parameters like quantitation, MS-MS tolerance, peptide charge, instrument, etc. in addition to fields for PMF and rest other parameters are similar to that of the peptide mass fingerprint. Depending upon the process carried out for data generation, a selection in the quantitation must be made. In case of label-free quantitation, select None in Quantitation tab. Remaining parameters remain same as PMF. Browse the MS-MS raw data file and search the results using Mascot. Define the instrument that has been used to generate the raw data. When we don't know the name of the instrument, select Default. The result output generated is almost similar to PMF output. The accession ID indicates the protein information obtained from database, marked as green. The protein hit outside the green box indicates p-value less than 0.05, which is statistically significant, whereas hits inside the green box indicates random matches. Mascot search results. Score. The peptide score is a sum of highest ion scores for each sequence with duplicate matches being excluded. Nominal mass. It predicted mass of the protein. Calculated PI value predicted isoelectric point of the protein. Sequence coverage indicates the percent of matching peptides. All the peptides are displayed with matching peptides indicated in red. The protein view obtained on selecting a particular protein link is very similar to protein view observed in PMF. It provides detailed information about each of the matched peptide displayed. The start end and end position of amino acids calculated and experimental molecular weights, number of missed tryptic cleavages, sequence of each peptide fragment and their corresponding ion scores are shown. The highest ion scores are used for computing the final protein score. Each peptide in tandem MS-MS undergoes a second round of fragmentation when it passes through the second mass analyzer before it reaches the detector. This provides significantly larger amount of information regarding each peptide fragment, which can be viewed by clicking on the peptide links provided in the summary report. The fragmentation pattern is displayed graphically, which can be zoomed into as per the requirement